Hello, everyone. Welcome to the third session for BGA24. Today we've got a very cool talk about annotating genomes the Ensembl way. I've dropped a link, some of the important links in the chat. The first one is a form, so I can gauge the people that are actually turning up to the sessions and we can improve it for the future. The other two are the GitHub page, which includes the Git pod, as well as the Discord link. When you fill out the form, it'll ask you for a code, so I can actually separate the information by session. And the code for this session is Ensembl. Right, guys, do you want to take it away?
Yeah, thanks, Damon. Let me share my screen. I think I need permissions for sharing my screen. Yeah. Hello, everyone. Has everyone able to hear me properly? I hope so. Yeah. Okay. So I hope you all are doing well. My name is Swati Sinha, and I work as a senior bioinformatician in the Ensembl Gene Build team. So today, we are going to have two sessions. In the first session, I'll talk about how we annotate genomes in Ensembl, followed by questions from you. And then in the second section, Jose will take a workshop for the RNA-seq data analysis. So I'll be going first. So let's begin. So let me first introduce you all to what Ensembl is. I'm sure many of you already know. Ensembl is primarily a genome browser, providing a single point of access for various genomes. We annotate genes, provide multiple sequence alignments, predict regulatory functions, and collect all sorts of disease data. As a user, you can retrieve gene and protein sequences, splice variants, and further annotation of genome at both gene and protein level. All the data that we have at Ensembl is provided publicly via FTP, REST APIs, MySQL dumps, and Biomart. There we have a dedicated outreach team which helps to take care of all the user queries, and they also provide training about how to use the Ensembl browser and fetch the data of interest at regular intervals. So do watch out for their announcement when they do these trainings. So Ensembl is also divided into various division sites, which provide data for specific clades like Ensembl bacteria, protists, fungi, metazoa, and plants. And they have dedicated genomes for each of these clades. We do have a rapid release website as well, which runs on a 2-week release cycle, and it deploys annotation for both non-vertebrates and vertebrates. We have more than 1,200 genomes for across more than 700 different species since June 2020. The current functionality of the rapid release includes gene annotation. You can have homologs from the website. There are repeat tracks which are provided, which can be viewed on the genome browser. Protein feature annotation. There is a utility to use BLAST on the server. We also provide HAL multiple alignments for certain clades. And there are variation data for specific species like human, fruit fly, and pigeon pea. But having said that, we are slowly moving away from rapid release into entirely new website for Ensembl that will include all the divisions, and it will have dynamic integration of all the data, full functionality. Basically, it will be one site to rule them all. So now let's get to the main topic of today, which is genome annotation. What is genome annotation? So in layman's language, the process of identifying and labeling information on a genome is termed as genome annotation. It could be coordinate-based, where you identify the different features on the genome, like repeats, genes, transcripts, exons, variants, and different regulatory regions. It could be knowledge-based, where you identify the function of these elements and effects of these elements on the phenotype. And lastly, it could be context-based, where different genomes are compared across different species to infer orthology, paralogy, and synteny. There are many different approaches for annotation. I have listed them here. The first one is the ab initio approach, which predicts the protein coding potential based on target genome and only. And it utilizes statistical models such as hidden Markov models. These methods are usually very fast, and the only input that is required is a genome. However, these methods can lead to a large number of false positives because these methods are particularly useful when there is no transcriptomic data. So these methods can lead to a large amount of missing or spurious exons. The next method is protein to genome alignment, which, as the name suggests, we align a well-characterized set of protein to the genome to infer the gene structure. These methods use existing data and they can give excellent coverage. However, since it's a protein data which is being aligned to the genomes, so usually they don't predict the untranslated regions or the long non-coding RNAs. The accuracy of these approaches can decrease if the evolutionary distance between the genome that you are trying to annotate with the protein set that you are using. And as you can see that this approach is highly dependent on the protein set which is available, and it doesn't really capture any novelty at all. The third method is projection and liftover, which means mapping the information from a reference genome. This can be highly accurate even at moderate distances, and it can leverage from the high quality reference annotations. The weakness of this method is that this could be highly expensive if you are using the whole genome alignment, and it doesn't really capture the novelty, as the genes which are covered, which are not present in the reference genome, will not be projected to the target genome. The last and the most useful approach is the transcriptomic approach, where you align the expression data to the genome. This is highly valuable, and we usually use the species-specific RNA-seq data that provides us with a better accuracy. So this type of approach is useful to capture novel genes and transcripts. It also helps to annotate the non-coding elements, and it can be used to help validate structures, gene structures which are identified through other approaches like leftovers or protein genome alignments. The weakness of this method is like it's difficult to get the high coverage of the entire full gene set because it depends on the expression data. Whatever gene is expressed will be used as evidence. Sometimes the sampling could be a problem and the cost could be a problem. If we are just using the short read data, this might also require additional inference for the gene structure. So in summary, what does this tell us? That we need an approach which combines different evidences to get the most complete annotation and the final gene set. And that takes us to how genomes are annotated in Ensembl. So Ensembl uses various different approaches. It combines evidence from transcriptomic, protein, genome alignments, projection, to come up with a final gene set for the target genome. So this is a diagram that just represents how the Ensembl annotation pipeline works. I'll quickly explain this, but I'll go in more detail of each of these pipelines further in my presentation. So basically, the first step that we do is we load the assembly and we do some quality checks. And once the genome is assessed for its quality, we annotate and mask repeats using repeat masker, along with the repeat modeler libraries. And we also use some other tools like DUST and TRF. We also annotate simple features on the genome, such as transcription start sites, CpG island with tools like CpG and Epunine, and tRNA scan using and tRNAs using tRNA scan. So after the initial processing, the actual gene finding is done through three main approaches. First, and the primary annotation efforts lying in using the transcriptomic data, both using long read and short read data. We align the publicly available data to the genome, but as I mentioned, this approach doesn't give us the full coverage and may end up with fragmented models or may have just missing unexpressed genes. So we fill these gaps using different approaches like protein to genome alignment and projection from a reference genome. And finally, any genes that are left are likely to be filled using the projection, which is leftovers from high quality reference genomes of closely related species. In the end, we combine all the models, do model filtration, remove any duplicates, and prepare a final gene set to classify proteins. So we'll go through all these in more details in a bit. But in general, what we do that when we have a publicly available genome assembly, which is not annotated, we look for the RNA-seq data, whether it's available or not. If the data is available and it's a vertebrate species, then we run our standard annotation pipeline. But if it's a non-vertebrate, we use a slightly different annotation pipeline, because for non-vertebrates, there is usually not enough transcriptomic data, and sometimes not a good reference is available. So we skip the projection, we use different protein sets, we rely on orthologs. And in case the RNA-seq data is not available, and we are doing a vertebrate annotation, we still rely on our annotation pipeline, but since the RNA-seq data is not available, so we rely majorly on leftovers and protein alignments. However, having said that, the available protein data which is present in the databases is skewed towards well-studied clades like mammals and other well-studied clades. So, and the leftovers we usually do from the GenCode annotations from human and mouse. So the leftovers and protein alignments may not be very useful for species which are distantly related to the target genome. In case the species is a non-vertebrate, we just simply provide a breaker annotation. All right, so let's begin with how we annotate genomes. So the first and the foremost important step in genome annotation is the repeat annotation. Finding and classifying repetitive DNA on repetitive DNA on a genome is very important, as they are a common feature of eukaryotic genomes. A recent work on the human T2T assembly developed an atlas of repeats that comprised over 50% of the genomes. So it is very important we annotate these repeats and mask them before any downstream analysis. And usually the masking and annotating the repeats are often done using the same tool. So there are different types of repeats which are present in a genome. The major ones are the low complexity regions like the polypurine or polypyrimidine stretches, or the regions of extremely high AT or GC content. We do have transposable elements divided into class 1 retrotransposons and class 2 DNA transposons, and satellite DNA covering the short and long tandem repeats. So why is it important to find and mask repeats? Because if these repeats are left unmasked, these regions can cause spurious alignments, providing false evidence for gene annotations. And if the genome is unmasked, it also requires a lot of computational resources needed to process the genome. So it's always advised to do repeat annotation before you do the genome annotation. So this is how the repeat annotation pipeline looks like in Ensembl. So given a genome sequence, we first check whether there is a repeat modular library available for the genome or not. If it's not present, we run repeat modeler and create the repeat modeler libraries. These libraries are given as input to repeat masker, which masks the genome based on the library inputs from the library and creates a soft masked genome sequence, which is then used for downstream analysis. In addition to running repeat masker, we also identify other kinds of repeats using different methods like TRF and DUST. And we annotate these repeat features, and these repeat tracks are provided on the Ensembl browser for user to check out where the repeats are present in the genome.
Bonjour, donc j'appelle, est-ce que vous êtes Madame Amanda? J'arrive pas à dire le...
Sorry, is that the question or? Oh, maybe it's. I'll just continue. Sorry. So repeat masking, as I said, is an important initial step in genome annotation. Although it presents a lot of challenges because it is computationally very costly. It takes a few days to get the repeat masking and repeat annotation done. The repeat libraries are generated from repeat modelers, and we don't filter them. So these libraries can sometimes contain gene families which needs to be removed. So we need to run BLAST against the gene family so that the repeats are the actual repeats. And identifying repeats in general is complicated as these are poorly conserved. The boundaries of repeats are sometimes not very well defined. And sometimes repeats can also insert within other repeats, and therefore the accurate detection of these repeat requires generating these repeat libraries for any species that you want to annotate. There are a lot of softwares which are available for doing repeat annotation, but there are only a few ones which are well supported. And if the only aim is to mask the genome, the recommended software is RED, which is Repeat Detector. And this is an extremely efficient tool, as I said, if masking alone is the only goal. So once the genome is repeat masked, soft masked, then we move to the actual process of gene annotation, which can be broken down into a number of approaches, and I'll go into all of these one by one. So transcriptomic approach, homology, ab initio, and the hybrid, combining all of them. So first, let's see the gene annotation using transcriptomic evidences. So the two major types of data that we use for transcriptomic alignments are the short reads, which are mostly coming from the Illumina data, and the long reads coming from PacBio or Oxford Nanopore technology. The short reads generally go through two initial phases. We align these short reads to the genome to find the exact exon positions, and then we reconstruct these alignment into potential transcript isoforms. For long reads, there is one initial phase and one optional. So we align the long reads to the genome to find the entire transcript structure because it's long read. So we can have the full transcript structure. And usually once it's aligned, we try to do the error corrections to get to identify the proper structure. And after the initial phases, both these data types have transcript models, which are usually collapsed to identify, to reduce the redundancy in the models. And then these models are used to identify the longest ORF, and once the longest ORF is identified, we classify these ORFs as coding or non-coding. There are strengths and weaknesses of both these approaches. The short reads provide better coverage. They are cheap, highly accurate, but can be easier to validate structure from other approaches. But they only infer the transcripts, do not observe them. So since it's short read, so we have to infer the transcripts, and UTRs are usually difficult to call. For long reads, it gives you the full length transcript structure. They are more accurate in identifying the untranslated regions, and they can be more accurate for spliced long non-coding RNAs. But in long reads, there could be lots of single exon transcription noise, which we do while doing the error correction. And it has been noticed that for ONT data, there is a high error rate when we use them. So there is some sort of minimal requirement if you want to have good evidence from the transcriptomic data. So the minimum thing that is required is short read data only. So if there is short read data available, then we go ahead with the annotation. And the short read data comes from different tissues. We have observed over the years of experience that the expression data coming from organs like brain, gonads, lung, gill, or embryo is highly valuable. And data coming from tissues or organs like liver, muscle, or blood do not provide that much value. The minimum length of these short reads, which is expected, is around from 100 to 150 base pairs, and we expect around every tissue to have 100 million, at least 100 million reads per tissue. What will be an ideal scenario? An ideal scenario will be that we have both long and short read data, so that we can combine the evidence from both these data. It's highly valuable if the data comes from more than 5 tissues, high value tissues especially. And if we have expression data from developmental stages, that's even more helpful. And the more reads, the better. It makes always a better annotation if we have more reads. And we prefer the consensus of clean reads over the raw reads. So this is how the transcriptomic part of our pipeline looks like. So we get the data from ENA for short read coming from Illumina platform. We align these short read data to the target genome using STAR, which is a splice-aware aligner. The output is a BAM file, which is then given as an input to Scallop. Scallop is a transcript assembler, and it assembles all the short read transcripts and collapses collapse into a transcript model coming from short read data. For long reads, we take data from PacBio and ONT platforms, and this data is aligned to the genome using Minimap2. We do error correction and filtering of the output, and these output gives us the long read transcripts, which along with the short read transcripts are collapsed and merged. Now, these transcript models are validated against the known protein coding genes, and this is done via Diamond, which is just used to align the transcript models to the experimentally validated protein sets, so as to infer whether the transcript model that we have predicted is valid or not. My presentation. So sorry about that. The next part is the gene annotation based on homology. So homology is when you compare one sequence to another in very simple terms. So we do cross-species protein alignments, as protein sequences are more conserved than nucleotide sequences. So this data can be used from closely related species for the annotation process. There are a vast number of available protein sequences with varying levels of evidence, like whether the protein is has transcript annotation, and whether it's experimentally validated or not. These proteins can then be used to do protein to genome alignment. So for example, this is just a pictorial representation. If your target genome is lemur, you want to take data from closely related species, in this case, say from human, chimp, gorilla, and gibbon. You take all the known well-characterized protein sequences from these organisms and align these proteins to the target genome to infer the gene structure. There are tools which are used for protein to genome alignment. We basically use GenBlast and MiniProt for these approaches. So the second part of homology is the projection from reference, where we use a reference genome and do a whole genome alignment. We generally prefer high quality assembly and annotation so that the target is relies on the reference annotation. Some of these methods focus on breeds, strains, or haplotypes and allow projection of coding and non-coding features both, while others focus only on the coding exons to allow projection across greater evolutionary distance. And this is just a pictorial representation of how a projection from a reference looks like. So the original transcripts are projected to the target genome using whole genome alignments. This is how our homology pipeline looks like. So we download the protein sequences from UniProt, which are at evidence level protein PE1 and PE2, which have protein evidence and transcript evidence. These proteins are then aligned to the target genome using GenBlast. We also use MiniProt to align the protein sequences to the target genome. Once these alignments are done, we create transcript models, and we also do, as I said, projection. So we compare if there is a reference genome available. We align this reference genome to the target genome using LASZ, which is a whole genome alignment aligner. And LASZ gives us genome alignment blocks, which are fed into Caesar2. Caesar2 is another method which basically realigns the coding exons back to the genome using statistical methods like hidden Markov model. Now, we combine these evidences to create a final transcript. So as you can see that we need evidence from protein, from long read, from short read to get the final transcript. And it's always useful to combine the evidences from various approaches to get a nearly perfect transcript structure. Okay. So what does it actually mean in terms of when we combine evidence? So if we have a lot of data, we will have like both long read, short read, and protein data. We will have more transcript isoforms. More genes will be captured per class, including time point and tissue specific ones. UTR's features will be predicted. We will have better exon prediction, especially around the start and the end exons, and we'll be able to measure the confidence of the gene set based on how good our initial data was. And if the data is poor, we don't really have enough data. We often focused on just protein coding genes. It could have just one isoform per gene, and we could miss some exons. There could be some misalignments, no UTRs, and there could be a large number of false positives. So once the genome is annotated, the next step is usually to assess the quality of genome annotation. And it's not an easy task. There are different approaches that can be used to assess the quality of the genome annotation. There are few things to look for, like one-to-one orthologous relationship with references using reciprocal best class hits. We look at the number of the protein coding gene counts and whether it makes sense to have that number of protein coding genes in the species that you're trying to annotate. We also look at whether the exons, whether the size of the exons makes sense, whether the average size is equivalent to the species which are already annotated in that particular clade. What's the CDS length? What's the genomic span? In addition to these little stats, we also use two different tools, BUSCO and OMAC, to assess the completeness for the genome annotation. So BUSCO, as you know, that it's a method to assess the genome annotation quality, completeness. It uses single copy orthologs from defined taxonomic clades. So what it does is Busco has already trained different clades and provides a set of hidden Markov models for each of these clades. So when you run Busco, you take your input, so it can be run in both genome and protein mode. When it's running in genome mode, it is assessing whether how complete is the genome, and the only input that is required is the genome and the lineage that it belongs to. So, but you can provide the lineage yourself, or Busco will assess which is the closest lineage to your target genome. And then it runs, it used to run MetaUke before, but recently Busco has moved to miniprot to predict the genes in the genome mode. In the protein mode, the input required is your annotation, which is protein sequences, and then it compares these set of protein sequences against the hidden Markov models for all the different lineages and assess how complete the gene set of your target genome. It provides a completeness score. So on the right-hand side, there is just a bar plot saying the Busco scores for different clades that we annotate. The other method that we use to assess the quality of genome annotation is OMARC. It is similar to Busco, and it estimates the completeness of proteome by comparing it to the orthologous group, which is usually defined as hierarchical orthologous groups or HOGs. OMARC assesses the accuracy and of the genome of the proteome, and it filters out error gene models in the proteome by comparing it to the gene families of the selected ancestral lineage. It also detects possible contamination, whether your protein set has proteins coming from, say, a very different clade, bacteria, or some other contamination. So OMARC doesn't really, unlike Busco, it doesn't really rely on a manual selection of reference data set. Instead, it automatically identifies the most likely taxonomic classification of the test proteome. So therefore, it can be used to process any test proteome set across the tree of life using a universal reference database. And on the right, again, there is a bar chart showing the OMARC scores that we get usually in our annotation. So to summarize of what all I have spoken today is that genome annotation is still something which is not totally solved. We still work on it and we find many different problems. But the general approach is that the first step is to do repeat annotation. And as I said, that many sources of repeat libraries and tools are available, but repeat modeler and repeat masker combination is the most popular approach. And then the genome annotation can be done by using a variety of approaches. At Ensembl, we use evidences from multiple evidences from transcriptomic data, from protein data, and from projection from a reference genome. So the quality of annotation will always be dependent on the quality of the input data used. So this is our team, the eukaryotic annotation team. I'm a part of it. Fergal is the team lead, Leanne, the project lead, and we have bioinformatician Francesca, Jose, and Vianey in our team. And I would like to thank the entire, apart from my team, everyone who works at EBI. We work so closely with all the other teams. Thank you, everyone. And thank you, audience, for listening to me. If there are any questions, I hope it was useful. And if there are any questions, I would be happy to answer them. Oh, sorry, I didn't realize I didn't turn on my video. Sorry about that. And I can't turn it on. Okay. Oh, yeah.
There you go.
Sorry, guys. I didn't realize it was off. So are there any questions, or was it useful? Did you?
I have a couple of questions, if that's all right.
Yeah.
Obviously, these are some pretty substantial pipelines that you're running.
Yes.
One How long do they take to run?
It really depends on the genome we are trying to annotate, on the cluster load and everything. But for a vertebrate, I would say 3 days.
Okay.
No?
3 days?
No?
No. Vertebrates require a bit more of work, and I would say.
Yeah,
some
I mean, in
research.
ideal scenarios, if there are no errors or anything, but usually there are errors in the steps that we are using. Sometimes repeat masking can take long, so
Yeah.
In ideal scenario would be 3 days, but practically it's like how much, a week? Yeah,
couple of
around a
weeks,
week. Yeah, a couple
probably,
of weeks. Yeah.
You have
Transcriptomic can be really
Yeah.
long as well if we are dealing with a species that has, luckily for us, a lot of transcriptomic information that can lengthen the process quite a bit.
Yeah.
So you have to fall back on like manually pushing things through the pipeline in the end, just because, well, I suppose you can't accommodate for all the variations in all the genomes. So that must be quite a pain in the backside.
Yeah, sometimes I like, because errors, yeah, because we'll expect no errors, but usually there are errors because of all genomes are different.
Yeah.
So there will be different requirements, say for aligning, star works for one genome and it doesn't work for other genomes. So we have to change some parameters. We try to keep the command as general as possible so that it's able to work for all the genomes, but in reality, it doesn't really work. So we have to modify a few parameters. So fixing all these errors in various different steps do require manual intervention, and I don't think we are getting rid of this manual intervention anytime soon. It's very optimistic as well because, as I said, the genomes are very different from each other. So we do require some manual interventions, but otherwise the pipeline is fully automatic. If it doesn't fail at any step, it just goes, it does all the step itself. So we just have to initialize it, yeah.
I understand a lot of the pipelines we build with the optimism of like, oh, this
Yeah.
one button to push everything. Nothing will ever go wrong.
Yeah.
Never the case, is it? And then my other question was, how do you prioritize what genomes to work on?
So we have mostly a number of grants or projects or collaborations that we tend to prioritize. Then on the different tier of priority, that would be so whatever the community essentially is interested in. So, for instance, say that a specific strain of sheep has become more widely used or is generally considered to be the new reference for sheep. So we will tend to prioritize that and have it ready soon so that people can use it. You are very well aware of the Darwin Tree of Life project going on, so that's obviously a huge priority for us as well. Whatever is within that project tends to be very high in our priority list. And yeah, generally speaking, that would be user interest and the very varied diversity projects going on at the moment.
Fair enough. Thank you very much.
Hello, I have a couple of questions, if I may. So,
Yeah.
I want to ask, how does Ensembl annotation pipeline compare to similar pipelines that use this combination of approaches? Recently, we have a very good experience with Annotate. So, have you made some comparison, for example, with Annotate and how if there's an advantage of one pipeline compared to the other?
Which pipeline did you say? Annotate.
Annotate.
Okay. So I mean, we don't really compare our pipeline with others. We just compare the annotation that we get mostly to the RNA-seq annotation. And we more or less we do similar things, but the pipeline that you have mentioned, we haven't compared our pipeline with Han and Annotate.
Okay, and my other question is, since Ensembl pipeline is supposed also to annotate the plants, and in the plants, like in most of the studies, the focus is on the repeats and not on the genes. Like there's some studies focusing on the genes, but most of the work I see is focusing on the repeats. But in the case of Ensembl pipeline, it only uses a repeat modeler, while as I see people working on the repeats annotation in the plants are using the combination of tools like repeat modeler, repeat explorer, a tied cluster, or a dumpty LTR, and then everything, all of the results are combined together and then are masked. So I can understand using the repeat the modeler for the vertebrate, but does it have a disadvantage for the plants in that way?
Vianey, do you have an answer for this? The most recently working in plants.
We do annotate repeats.
We do annotation of plants, but using the other pipeline that just use basically transcriptomic data, and also the repeat annotation is done. And so far we have obtained good results. So we have annotated some flowering plants. So with those have worked well the current pipeline that we have.
Yeah, so we do annotate repeats and use the transcriptomic data set for plants as well. And I think some of these are handed over, right?
Yeah.
So some of the recent plant annotations that we have done are already available on the Rapid Release website. So if you may want to look at them, because they will have the repeat annotation, and we have used the transcriptomic data, whatever is available, and we have been able to get good BUSCO scores for these plants as well to assess the gene quality that we have predicted.
Okay, thank you.
Thank you, Hassan.
There's another question in the Discord from Sam. Two other
I'm taking care of that.
Oh, you're taking on it. Okay, cool. I'll leave you to it.
Sam, what do you mean with your first question?
Yeah, I was just curious on how you're defining the one-to-one orthologs, because that can be, I guess, somewhat problematic. What kind of thresholds or scores are you using to determine a true ortholog, I guess?
From which part?
This is for after we get the annotation, so to assess the quality, sometimes we can check by doing a reciprocal blast hit to assess the one-to-one orthologs.
Okay.
I'm
not really familiar with that part. So
yeah.
So we'll look into this and we can add it to the document later.
Documentation later, yeah.
We'll compile every question you make, and even those we can't answer at the moment, we'll try to answer somehow, and we'll share this document with you later. But
usually, I can talk a little about it, because if we are using BLAST, so we usually rely on the what kind of coverage and score we are getting for that particular gene. So if the coverage is good, if the identity is high, we usually can say that it has the one-to-one relationship. But obviously, I can go back and add more to that question.
Yeah, I guess I'm just curious on the overall threshold. If you're saying a 1-to-1 is a 70% like match through BLAST P or something like that, or if it's 80%, or because there can be some divergence, especially between a haplotype set, I guess.
Yeah, so for 1-to-1, it has definitely to be greater than 80-85%. Definitely, that's the threshold.
Thank you.
I don't know I don't know the exact number yet, but we can find it out. Yeah.
Okay. Well, you can keep posting questions if you have them in the Discord, but maybe we can then move to the next bit. Which one?
Yeah.
It's
You can ask them if you want to take a break or something.
Okay.
Oh God.
Can you hear me now?
Can indeed, yeah.
Okay, good. Okay. So next we are going to do some exercise, which will guide you through some of the steps to deal with the transcriptomic information we often use. But I was wondering if maybe it's a good idea to have a very short break just to grab some coffee, tea. So let's say we're back at 5 minutes to 2 British time. Is that cool? Cool. Then we'll see you back in a moment. Thanks. Okay. Yeah. Demon sent before a link to sorry, what is the page, the GitHub page, but no, where is this, sorry. I will share this session's link, which if you access, you will find the GitHub button there right underneath this session is part of BGA24. That says open GitHub, open Gitpod. If you click on that, and well, let me share my screen already. Okay. Can you see my screen?
Yes.
Well, so I make sure that the last option says large, not standard, because that will help us a bit move things smoother. Okay. The rest, the first field, don't touch that one, and this is absolutely up to you. So you can click on the comments, but then forget about it for now, as I will talk a bit before that. Should I start them on, do you think?
All good, yeah.
Good, awesome. So yeah, as I said, this is a very short exercise, like a bit of a mock of an annotation, or specifically one of the steps of the annotation we perform. We are going to try to turn some RNA-seq reads into gene models. It's a good thing that Swati has explained you all about our pipeline. But we are going to be focusing only on a little bit of it, the RNA-seq pipeline, which, as I mentioned briefly before, is one of the longest ones we have to run, or one of the longest parts, mostly because, as you are probably aware of, RNA-seq data tends to be very computationally heavy to work with. So in this approach, we are going to be using some data that we have used many times before and curated until we have a quick to use, quick to run set of data that yields some interesting results and observable results. The main point of it is that you can see the immediate result of a very quick analysis. Okay. In that sense, in the Gitpod, I've provided a genome and some short reads. We'll run a start on them, sort the bound files, run a scallop, and get some GTFs. Finally, in a normal annotation, although we won't be doing this here, we would build the final gene models and finally identify which of those are protein coding. Again, we will not be doing these last two steps today, but we'll discuss them very briefly at the end, so I'm mentioning that. So I don't want to waste much time here, but just in case someone is from more a computational background, a very, very short introduction of what transcription on RNA-seq is. Essentially, when all the information, all the biological information is packed into our genome, which is read, transcribed into messengers that eventually get analyzed and transformed into the final product of our genome, which is going to be proteins. Very briefly, okay. What are we going to work here with? Or rather, what's RNA-seq? So we will take all of these intermediaries from a given organism. Most often we will separate between different tissues, between different developmental stages, for instance. So yeah, essentially what we are capturing is what is a specific cell or a specific group of cells working on at a given moment. We sort of freeze that into a library of reads, short reads, that we can then process and see how they would have behaved. So we can align them to the genome to see what were they processing, for instance. We can infer the final product of this. We can assess different levels of expression of different genes, thus understanding a bit what goes on in a specific moment of the lifespan of a cell, for instance. In this specific example, I've given you two pair reads for two reverse, two forwards for two developmental stages of the zebrafish, specifically when it is two cells and Let me find it. Sorry. Well, sorry, I cannot find it in the schema, but yeah, it's a different. Oh, sorry, 6 hours per fertilization. So 2 different stages in the very well-known developmental stages of the zebrafish. Also, we're providing the genome that we need to align them to. Again, to lower or lessen the computational needs of this experiment, we are only using 1 chromosome because we know for a fact that the window we want to check in the genome is in here, and we will have the results. Finally, when you go back to Gitpod, which I cannot do because I share. Okay, I share the screen incorrectly, but I will fix that in a moment. When you go back to Gitpod, which you can do, you will see that if everything went correctly, Conda should have installed some software that we are going to be using. Conda is essentially a library manager that will allow you to handle a lot of different software. And it's an easy and quick way of getting ready to work, let's say, software for biology in particular, very used. No, sorry, let me stop share. Share desktop too. Okay. Now, yeah, you have the Gitpod as before. Remember the options I mentioned, specifically this one being last because that will help with greatly the exercise being smooth. And then my personal preference is to open this in a browser, but you will be able to open it in PyCharm, in Visual Code Studio, whatever it is that you have indicated on here, okay? So if everything goes correctly, I already opened this to do some tests, so I have everything ready, but you should be seeing some messages here. Those messages are essentially Conda working its way into installing everything. So can I have some thumbs up if you are seeing that? Good. More people. Are you all with me? Good. This is much more of on the hands exercise, so please feel free to unmute yourself and just shout out if you're behind, or if you missed a step, or if you don't understand where we are, or you see something different that I am telling you you should be seeing. Okay? So just much more casual everything. But yeah, if no one else says anything, I'll assume all is going fine and move on. Now, I guess maybe you have used Gitpod already in these BGA talks, but in case you haven't, and this is your first time, you will have a list here to the left with different buttons. Most importantly, these 2 papers are going to let you explore the different files. And I'd like you to see if you can access Ensembl annotation, and if that's the case, see if you have an Annotating the Genome folder. Furthermore, you should see, much as I am showing, the 4 FASTQ files I mentioned, 2 for 2 cells and 2 for 6 hours post-fertilization. And finally, you should see the genome that we are going to work with, which is too big to open, so let's not bother with that. The genome I haven't mentioned, but it's in FASTA format, so pretty standard. I'm sure you have worked with that many times. Good. Let's keep moving then. This is more or less covered. Minus this file, which you should be able to open. It's a markdown set of instructions. So it looks a bit weird here, but if you opened it in a markdown supporting device such as GitHub, it will have a much more readable and pretty structure. Well, doesn't matter much because we are going to mostly copy some stuff and see how that goes. So yeah, let's start from the very beginning. So step 0. So that everyone is in the same page, I'd like you to make sure that you are in workspace Ensembl annotation. And for doing so, we are using 2 extremely basic Unix Linux commands. First, we are changing our directory and then printing which is a working directory. Everything seems to be fine for me. I hope the same is to you. And again, please interrupt me, just shout out if it isn't. And yeah, let's have a look. Again, you will notice that this is exactly the same thing we are seeing in the left-hand panel, only with some extra information that we don't particularly care about. So let's ignore that for now. Cool. So first thing first, we are now going to use STAR. STAR is a software that maps short reads to genome. STAR is a good choice for this because it is able to map over splices. It has some degree of tolerance to mismatches. And also, it is built into it to avoid the unwanted sequences, such as, for instance, the poly-A tail. Okay. The only thing is that, as practically every software that maps something to something, it requires an index of the genome we are about to map. So before we go and do that, which is in the instructions, I just wanted to mention that STAR website is, well, the GitHub page specifically is pretty good, has a very comprehensive tutorial and docs, and yeah, please check the on-site, obviously, the software if you ever use it. But now let's go ahead and index the genome. For that, we are going to use STAR followed by the run mode we want, which is genome generate, and then the information it needs, such as where the genome is, where do we want the index to be stored, and so forth. This is a pretty straightforward command, so I don't think we need to delve much into it, but again, please interrupt, ask questions, and let me know if you want to know anything else. But yeah, you will see something like this, or hopefully quite similar to this. We are having a sort of log that is informing us of the different steps the software is making. No? At the end of it, we should have several new files in this genome folder that I mentioned before. And if you keep that open, you will see them appearing in real time, which is very useful for these sort of exercises. So yeah, we have several text files and several log files, index files. All of those recently created, that's the green color. And those are essentially a guide that the same program star will use when we tackle the next step, which essentially involves the actual mapping. Now, let's start the next step because it may take a while and I will talk to you in between. So as we are dealing with two different tissues, we are going to need to run star for both things. It allows the pairwise, so when we indicate the files we want start to process, we can indicate the pair. So we'll be doing forward and reverse. And then we will be doing again forward and reverse. We are requesting specifically that the output of the result is in BAM, and we want a sorted BAM, sorted by coordinates. The other parameter is only the name we want to give to this BAM. Now the first one is done, and you will notice that there is a new folder called BAM, filled with several different output files from this star. All of them from 2 cells, as that is the one we just ran. We will now repeat the same for the other pair, the 6 hours post-fecundation. And we can maybe now pay some attention to the standard output we get, which is, again, pretty straightforward, a log of time and what the software is doing. If you go over it, it's pretty straightforward. Logged in genome, start mapping, finish mapping, sorting BAM, finished. I will take this chance to check that everything is with me. Are we all running the second pair 6HPF? Good, thanks. Okay, good. Then we have now created a BAM. Now again, what's the BAM? The BAM will be the alignment of our reads to the genome. Okay. SAMtools is mentioned here because it's a very useful suite of diverse software to manage BAMs and to manage SAM. So it's always useful to have. I think we will be using it later. But yeah. Oh no, sorry, we are using it now to get up. Okay. Apologies, I forgot this step. So yeah, we are now going to use SAMtools, which is again a suite of software suite of diverse and very diverse, as you can see here. This is the list of all the different subprograms or modes that SAMtools have. And we are going to do this just to check basic stats on the end product of our previous commands, okay? This is something that, we do more for you to see here. We don't have them in the pipeline we use as it is here. We do a lot of quality check, but way different. In any case, I think this is a very useful software to know because, as you can see, in a matter of seconds, you can have a lot of very interesting stats. Specifically, you can see that 100% of our reads were mapped, which is a very good thing. You can get an estimation of the number of reads per file. Again, a very reassuring 100% of properly paired. So essentially, all of these looks great. So we can move on. Just so that you can check again how quickly this is the second pair, which again looks the second BAM, which again looks quite nice. 100% aligned, all proper pair, yada, yada, yada. And now, yes, the important bit. We are going to use scallop. And what is scallop for? So we have now this status, no? We have several reads aligned to the genome, essentially. But we can get information only from that alignment, of course. But it's so much more useful if we can have some tool, such as scallop, to provide predicted models. So scallop is going to take these reads, these aligned reads, and is going to try to find the patterns that define different transcripts in there. Okay. This should be pretty quick, but let's go with it. Super quick. And again, you can see that a new folder appeared, or should have appeared. So, oh, no, sorry, because we are, again, indexing first. So the only thing that will appear now is the index. In fact, this cannot be more simpler of a command. We are asking some tool to index this one and this one. Okay, again, once per different tissue, or in this case, developmental stage. Sorry, this is the folder I was looking before, which we should create. This is a Unix command to create a new directory. In this case, we are calling the directory GTF. And if we go here, we can see that it has been created. It's empty so far, but I think we'll fix that in a moment. By running this time, yes, proper scallop. First, we are building models for the choose cells stage. And you can see we are providing the index that we just created in the previous step. We are giving it an output name. And then scallop is very noisy in the sense that there is a lot of standard output messages, very long logging, which is a good thing if you encounter any issues. But it's difficult to go back to if it's just printed to the screen. So we are using this pipe to just send it to a file. So if we check now GTF, yeah, we should see both the result of our scallop, the GTF, the standard out. You can see plenty of information, which again, unless something went wrong, you don't necessarily want to waste a lot of time reading. So we'll leave that for now. And of course, you will most likely be familiar with the GTF format, which is essentially a listing of features sorted or defined by the coordinates and the parents they have. Okay. But yeah, without much more, let's run the second BAM scallop. So this is for the six HPF cells. No one is complaining yet, so I will assume that everyone is on the same page as me. Sure, it has finished successfully for me. We have the GTF here, and we have the standard out here. Let's clean up a bit the view. And yeah, now if you all confirm that you have finished with that, we will start with a more fun part. That is, of course, reviewing the results. And for doing that, since we are representing Ensembl, we thought what better to use Ensembl browser, which hopefully you know how to access, but if not, I have included sorry, references are now. Yeah, I have included a link there for you to use. There is also this other link that you should have at hand. Okay, because we are going to use the Ensembl browser. You need to provide, so one of the many advantages of the browser is that you can provide your own data and analyze that on top of the data we provide, which is what we are going to do now. The only issue is that you need to provide the data, and there is no extremely easy way of getting this data from Gitpod that I am aware of. So instead of going through the trouble, I have uploaded beforehand everything in this FTP directory. Okay, so if you access this and remain in this site, in this view, we'll use these links in a moment. You can find again all the steps here. Okay, so let's now search for this gene, which is the one we are going to be using for the visualization of this Bambi A. Okay, this should be what you will see, a very basic view with just the gene and little else. What we want to is to add some more info to that. Okay, how do we do this? We go here to add remove tracks. Now on the top, on personal data, and now we name it. So we want, for instance, 2 cell and 2 cells. Bam. We go to the one I provided. Bam, 2 cells. And you copy the BAM one, not the BAI. Okay, this is the index I was telling you before. Now, the magic of the Ensembl genome browser is that if you provide a URL with a BAM, and in the same URL there is a BY, you do not need to upload anything else. It will read everything, and it will, or should work fine on its own. Okay. Now, I hope you were able to follow me, or that if you weren't, you tell me right away. But I will be.
Sorry, I wasn't able. Sorry, I lost it when you were starting to add the feature. I was looking at the other screen at that moment, so I don't know where you clicked.
No worries. I have another 3 of them to add, so let's go this one again a bit slowly. So
no, even one step before.
Oh, sorry. Here.
Yes.
Okay, so this is the stable ID of the gene we are going to be watching. So in this top right corner, you can search for the, well, not for this. You search for this, and it should take you directly to this website. You will not be seeing this or this, because this I just added. So you should only have these genes,
yes,
contigs, and that's all really. Okay, good. So then you go to this add remove tracks. Alternatively, to configure this page, both of them are the same. So you go here, and on the top left, you have configure page or personal data. So you click on personal data.
Okay.
And then you should have directly this. If not, you will see as before, like add more data, no?
Okay.
Yes. So bear with me as I add everything, and
thanks.
Feel free to keep, essentially copy the BAM files from the URL I share with you and paste it here. It will immediately detect that it's a BAM, and you can add it straight away. from here, it's just like, thank you, remote data was successfully attached, yeah. But you can click on custom tracks again, and you will be taken here. Okay. We did BAM for both developmental stages, so now let's add the GTF as well. Okay. 2-cell, 2-cell GTF. Now we need to go to the other folder, the GTF folder, and copy link. It has automatically detected that it is a GFF2 or GTF. So we add it. Thank you, file upload. Good. Custom tracks again on the last one. So, GTF. We copy the other one and we paste it. Good. We can close this window or this pop-up in the top right corner in the tick symbol. It will load for a while. Depending on the usage that we are getting at the moment, it can take more or less. So give it a time. And then you should see something like this. You can rearrange things by just dragging them. Okay. And then, finally, if you go back, sorry, I'm going probably too quick. If you go back to configure this page, you will have at the very top of active tracks your data, and you can select what sort of visibility you want to have. Okay, I am going to pick normal in one of the BAMs, unlimited in the other so that you can see the difference, and then both of the GTFs in normal. Okay. Once you have done the changes, again, save. And now it didn't work. Why? Oh. Are you seeing something? Okay, good. Well, let me know if you can see this in your own computers, if you are doing this parallelly to myself. If not, hopefully you can see it in my screen at least. Good. So, yeah, let's zoom in a little bit. So if you click somewhere wide and drag it around, you can then jump to region view, and you can have a slightly more detailed view. Okay, so I'm doing that, and again I am going to move this up. Good. I don't think we need these, so we'll move them down. Okay, so this is, by the way, when you upload a track, it's loading for the forward and reverse reads of the genome. So I'm not using these ones because these are the incorrect ones for this specific gene, okay? This seems to be a forward gene, so this must be the reverse interpretation of the data, which is a bit meaningless. Let's stick to this one, which is the more interesting one, as you can see, hopefully. So what do we have here? You can read the name of the tracks on the left. and we have the 6 HPF GTF, 2 cells GTF, which are quite different, as you can see. We have the 6 HPF BAM, and oh no, sorry, I discarded a correct one. So yeah, let's get rid of this one. Seems to be quite uniform, so it's fine. Yeah, so we have the 6 HPF BAM and the 2 cells BAM, and you can see that they are both still different. You have to pay a bit of attention, but you can see next to the name is the scale, and if you look at that, you will realize how much more remarkably different they are than you probably thought initially, as this is on a scale of 0 to 1,000, meaning that this is probably close to 1,000, while this is on a scale of 0 to 14, meaning that this peak here might be 14. So essentially, this dark gray is the coverage. Okay, so we align the reads, right? And you can see them here stacked. These are the reads. The green bit is the beginning or the end. I am sorry, but I am not entirely sure at the moment. I don't remember. Okay, yeah. Never mind this. Yeah, so the green bit is the beginning, which means that this is the forward read, so this is the one we want. Sorry, let me start again. This is the gray part is the coverage. The dark gray bit is the reads themselves aligned. Okay, you can see the traditional RNA-seq stair-like pattern cascading. And this is the coverage. It is still huge, actually more huge. It's 1,200 at its peak, which means that this particular region of the genome has that many stacked reads, essentially. And then we can see where the reads align. Same goes for the 2 cells, and again, notice the huge difference. So this is one of the things we wanted you to see today, like very quick usage of little amount of data, and you can already tell how greatly different this specific gene is treated in 2 cell stage and in 6 HPF stage of the development of this species. The BAM and the coverage is a bit more tricky to interpret, but essentially the take-home message is that these GTFs, if you remember those that we created with the scallop, are the different interpretation that scallop has made of this. Okay, so you can see here quite a bit of a difference. For instance, this first exon seems to be much more smaller in the 2 cells than the other one. This exon seems to be also much longer in these isoforms. Here, Scallop has found what could be possibly a monoexonic model, which will likely not be a true one, but we'll get into that later. Yeah, 2 cells, for instance, seems to be much more consistent. The 2 isoforms must be practically identical, maybe some nucleotide differences, while there is much more variability within the 6 HPF GTF. That, combined with a much more presence of transcriptomic, is a clear indicator of, well, I shouldn't say clear, it's an indicator of a much important expression of this gene in this stage. Now, this is some results that you have to take with some care, because for these results to, in terms of expression, in terms to be truly significant, you need to have some, oh, sorry, I'm missing the word in English. Some control that you know is quite ubiquitously expressed. Acting, I think it's one of the most used for this. And you need to normalize your levels when compared to that acting levels, okay? I don't know if I made sense. I hope I did. But yeah, this is merely an indication. This would never be taken as a core truth until further researched. But yeah, but this model, gene model generation, this is something that we do take and use. So as Swati explained, this will be taken by our pipeline and will be essentially combined with the other results from other parts of the pipeline. And what will emerge from that is a hopefully quite curated and robust set of genes. Yeah, this is what I was just showing you. And finally, yeah, I mentioned at the beginning that this is not the end at all. It is the end of the exercise, but when we are annotating proper, once you have created all of these models and you have filtered through those, for instance, you want to filter through those, so for instance, getting rid of this likely incorrect scallop presumption, no? So in order to do so, one of the things you want to do is try to identify which of those transcripts are protein coding, which aren't. This is an extremely complex issue, and an issue that I don't think is yet fully solved. But we are seeing a lot of new software that promises to work pretty well. And we have some solutions in place, but we are constantly trying to find better alternatives as possible. So yeah, as I was saying, RNA doesn't necessarily mean need to be messenger, meaning that will eventually probably become a code. Can be small non-coding, long encoding, ribosomal, microRNA. So many different alternatives, and we need to differentiate between them because it will affect the annotation, but I want to clarify that all of them are quite useful and all of them play some role, some of them increasingly known and important roles. So it's important to differentiate all of them. Sorry, yeah. One of the alternative software we use is Ensemble, which predicts the possibility of a transcript to be protein coding, and thus will be classified as that and be taken into consideration for the creation of the final gene set group. But there are alternatives to this that we are exploring. So I just wanted to show this because yeah, it's a bit old, well, relatively old, but it shows a comparison of different alignment software for RNA-seq in Arabidopsis, I think, yeah, Arabidopsis thaliana. Yeah, all of them are quite well performant. Star strikes as one of the best, but yeah, HIS82 is also quite good, and we have used Salmon at some times because it has some advantages versus Star, specifically the speed. But I think Star still outperforms because it deals better with splicing. Yeah, that's about it. I have noticed that there will be some Nextflow content in this BGA as well. So I wanted just to mention this Nextflow NF-core pipeline, which is designed to deal with RNA-seq data, essentially, in many, many ways. So if it has everything sort of ready to be used, so could be a good alternative if you don't want to go through all the hassle of having to configure yourself your own environment or get your own software. If you are familiar with NF-core, maybe this is a good alternative. And yeah, that's what I wanted to talk to you about. I hope you didn't find it too tedious, and that even though it was pretty simple and basic, I hope it's useful somehow. This is my team, as Swati already introduced, and the whole of Ensembl. Yeah, thank you very much for listening and following through. And if you have any questions, again, feel free to ask, type them, or whichever method you prefer. Oh, there's a question from Chiara. At this point, what would really make us happy is to have RNA-seq data that is accurately or methodically labeled, because I think the biggest issue that we have with RNA-seq data is really identifying the data itself, if that makes sense. Dealing with whatever the data has later or pre-process it for an annotation is fairly easy, but the lack of probably strict guidelines on how to name tissues, for instance, is our biggest issue. Because you will find the same exact tissue maybe named differently, or you will find a sample who named like male, and yeah, no more information provided. So like, how do you do with this then? And that's something that you cannot fix in pre-process or anything. The information is what it is, and you either chase down whomever submitted for more information, or you are really have to deal with that. So I would say that's our biggest issue, and what would make us really happy on our jobs a little bit easier is to have more systematic approach in naming samples and biosamples. Also, to give some more information, one of our main projects at the moment involves precisely this, trying to get ahead of the availability of an assembly and start to prepare RNA-seq data the moment it's available, and then have it ready to run essentially and store in our own, the metadata of the data stored in our own table, so that it should be as easy as to say, oh, there is this new ship assembly. What data are we aware of from ship? All of this. Okay, so, and all of this is ready, so it's just a matter of hitting a button, as we could say, and let it start. So that huge pipeline, again, still in development, involves a lot of this quality assessment, pre-process, preemptive alignments, yeah, a lot of analysis. So hopefully that will also help us a bit shorten the time that the transcriptomic pipeline often takes. Anything else? Good. Well, if there are no questions, well, I should say, feel free to contact us. If you access Ensembl website, you should see a help desk link. Whatever you ask there will reach us, and we try to keep up to replying this as soon as we can. So if you have any question, feel free to use that way. We will again compile all of these questions and comments into a document and make it available to you through Daemon, along with the presentation from before. This presentation is already on the GitHub, so feel free to just drop there and grab it if you need it.
Well, thank you all for coming, and thank the three of you for running a session today. It's been very interesting, and hopefully you come back and do it again next year.
Yeah.
Cool.
Thank you very much, Damon.
Thank you all, and yeah, goodbye.
Thank you.
Bye. Thank you.
Thank you.
Bye.